Right, grade 10s, this is the last little task that we need to do before midterm. Yay! Okay, so let's mark the photo worksheet of the different tissues that we've done up until now. So this is for lesson 6. All right, so I'm using the color photographs, and I think it would have been easier for you to do the same because black and white is very difficult to see. And so this first photograph, we have to identify each plant tissue and provide labels where applicable. All right, so if you look at this, there are the guard cells. Did I label? Oh, yes. So there's a guard cell. Well, that label's maybe a bit long, but this is an epidermal cell, and this has been stained. Okay, the cells of the epidermis on a leaf are really not this color. And remember, only the guard cells contain chloroplasts, but the epidermal cells themselves will be colorless. So I've labeled it leaf epidermis, and I've labeled the opening there a stoma or a pore. And remember, the plural is stomata. So there, 1, 2, 3, 4 stomata, and it looks like that one's still closed. So transpiration and gaseous exchange can occur through the stomata. All right, and there's a nice scanning electron micrograph showing the guard cells, although you can't really see that there are 2 of them here, okay, but there would be a line, a cell wall going down there. Almost looks 3-dimensional. All right, then I asked you to label C. We haven't done this tissue yet, so we'll do that at another stage. Right, so now we've got these little things sticking out, and that's root epidermis, okay, and that's a root hair cell. Now, one thing I didn't mention in class, if you want to, you know, when we grow seedlings from seeds, you plant the seeds in the ground, and then when the plants, you know, they might be a little bit crowded and you want to take the seedlings out and transplant them so they're not so crowded, it's actually quite important not to just pull the plants out, but that you actually take some of the soil with the root, or else you are going to remove all these root hairs, and that's going to result in the death of the seedling because now it's got no extra surface area and there won't be enough water and salt absorption. All right, so that's a scanning electron micrograph, and these root hairs are 1 millimeter long. They are really, really tiny. We're not talking about the big branch roots that would grow out of the side of the root to anchor the plant. Okay, so if we then look at another photograph, this is a photograph that was taken with a light microscope. Right, doesn't look as 3-dimensional. That's a scanning electron microscope photograph. This is light microscope. So there's the root epidermal cell, and it looks like that is the actual root hair. And here's another one, and they're unicellular. Okay, can't always see exactly where where it's from the epidermal cell because you're looking at the root from the side. All right, so that's photo C. Okay, so remember we're practicing to be botanists here, and I do have prepared slides of these tissues, and hopefully we can look at them, all right, using the microscopes. Now this is the tiny, tiny tip at the end of the root. So there's the meristematic tissue, and you can't really see that it's well defined. At a higher magnification, you would be able to. So that's the meristematic tissue, and this is the root cap. Okay. Then photograph E. Now this is my favorite one, as I said, parenchyma, the packing tissue. Looks like bubble wrap. You can see these tiny little triangles. It's exactly what it looks like. The cytoplasm, you can't really see. It hasn't been stained for. Now there's the nuclei, etc. And sometimes parenchyma can contain chloroplasts, like in the leaf or around the edge of a green stem or near the edge of a green stem. And the cells can have many different shapes. So on that PowerPoint that I showed you in class, some of them were oblong cells and you cut them in cross section and they're round. But here we've got some really odd shaped cells, but they would be much longer if you looked at them from the side. And then lastly, photo F, so it's parenchyma with huge air spaces. So I've called it aerenchyma. So that's parenchyma that's found in a water plant with these really, really big air spaces. And then obviously these are the cells, there's cytoplasm, and some of the cell contents can be seen. All right, so that's a little bit of botany. And when we come back after midterm, we're going to start recognizing the strengthening tissues and the vascular tissues. All right, and that will be the end of plant tissues. Okay, have a good midterm break, everyone. Stay warm, keep safe, and wear your mask. Bye-bye.